want to thank you. You're going to hear some things today about fibroblasts. And just to right up front, there's 2 cells in the human body we can use to regenerate tissue and cure chronic disease. There's a stem cell, which everybody's heard about, and there's a fibroblast, which nobody's heard about. It's the most common cell in the human body, so you'll hear a lot about fibroblasts. You'll hear the word homeostasis, where we're trying to take the body back to its healing zone. So we're excited with what we're doing. And when I say we're doing something unique, and I'm sure as analysts, every company that presents to you says we're doing something no one else is doing, we literally are. We can't find another company anywhere in the world that's using fibroblasts therapeutically. Our leadership here today, we have decades of experience in product development, clinical trials, capital formation, a lot of work in patents. Our board of directors, these guys are all in the game. When we set about putting together our board of directors, we wanted people that were currently in the business in pharma, and so they're experts in product rollout, clinical trials, capital formation. We have one rocket scientist, Richard Salento, on the board, so when someone says it's not rocket science, we all look at him. Our scientific advisory board, when we were putting this together, we went out to look for the world's foremost experts in fibroblasts. They didn't exist. You'll hear Hamid Khoja, our Chief Scientific Officer, talk later today. He is the world's foremost expert in therapeutic use of fibroblasts. There's no one that has the base of knowledge and the science that he's developed. So we went to the stem cell world, and the people on this list are the world experts in stem cells. When they did their research and they thought about the capabilities of fibroblasts, they couldn't wait to join our board. Our most recent one is Kate Rubins. She's an astronaut. She's the first astronaut to sequence DNA in space. It took quite a while. NASA doesn't really allow them to do things outside of NASA. She was so interested in this that she fought to get on this, and we're excited to have her. Some of our highlights, we'll be initiating a phase 1/2 clinical trial in the next couple of months for our diabetic foot ulcer. We'll be filing in our IND later this year for multiple sclerosis. You'll hear about our IP platform. We have 160 plus patents. That means we have more patents in fibroblasts than the rest of the world combined. So it's truly a space that we own. Again, our experienced management team, and we've just completed our master cell bank and our working cell bank that will feed into our drug product for our clinical trial. Why do we like cell therapy? This is the core of everything that we do. We've done the research, and we have not found any chemical compound in the history of big pharma that's ever cured a disease. I mean, think about it. Think of the trillions of dollars we've spent since the 1960s to develop a chemical compound to treat and cure a chronic disease, and it's never happened. After 60 years, it will never happen. It's not designed, we're not designed to have an external chemical compound cure a biologic defect. It will take a positive biologic process to cure that defect. So cures will come from cell therapy, gene therapy, and immunotherapy. And when we talk about fibroblasts, and people will ultimately say, well, if they're so great, why don't we know about those? Our theory is, and you'll see it in cell therapy, gene therapy, and immunotherapy, is that the Nobel Prize is a catalyst for the next decade of development. So take cell therapy, for example. Dr. Yamanaka won the Nobel Prize in 2012 for an induced pluripotent stem cell. It kicked off 10 years of new stem cell companies forming. The irony is that he used a fibroblast to do that. So fibroblast, our core cell, actually kicked off the stem cell revolution. After him, Jim Allison won the Nobel Prize for immunotherapy for cancer, and you saw 8 to 10 companies form in that space. And most recently, Jennifer Doudna and Emmanuelle Charpentier won the Nobel Prize for CRISPR, and you see gene therapy taking off. So when we want to cure a biologic defect, it's going to take a positive biologic process. While 30 plus companies are out there in the stem cell space, we are the only one doing fibroblasts. We like fibroblasts. They can differentiate into a multitude of other cells. The interesting thing about fibroblasts, they're fully differentiated, so they are a stable cell. Stem cells want to be anything other than what they are. So they're inherently unstable. They're inherently difficult to work with. And fibroblasts have a higher immune modulation. They have higher, more potent therapeutic attributes than stem cells. They actually can recruit stem cells in to help in the healing process. They're more common than stem cells. They actually outnumber stem cells 5,000 to 1, so they're the most common cell in the human body. And they're a universal donor. My fibroblasts work in you just as well as your fibroblasts work in me, just as well as my current fibroblasts work. How does it stack up? They're less invasive to source. They are, again, the most common cell in the human body. They're easier to culture, easier to maintain, easier to ship, easier to work with, and they will always be a lower cost alternative to stem cells. Some of the areas you'll be hearing about today are diabetic foot ulcer. You'll see some charts. You'll see a lot of science around this. What you'll hear is that we heal in half the time of the #1 product on the market, Graphics by Smith & Nephew. Multiple sclerosis. You'll hear from Hamid a concept of homeostasis, where we can take that area back to homeostasis and allow the body to heal itself. And we've seen remyelination in the healing of that brain tissue. Psoriasis, a very exciting area. We have some really exciting science on that. Degenerative disc disease. You'll hear in our thymic regeneration program about an organoid. That's a collection of cells that perform like the larger organ. And Hamid and his team have been able to create a small organoid, inject it sub-Q, and it stays durably fixated for over 60 days, producing T cells. So really exciting. If we are successful in our thymic regeneration program, we can keep humans alive another 10 or 15 years, because what happens when we get into our 60s and 70s is our thymus gland stops functioning. We don't have more cancer at 70 than we have at 40. It's just that our thymus gland stops teaching the immune system in our 60s and 70s. If we can take that immune system back to a functioning immune system, you not only will extend human life, the quality of life will be much better. We have some exciting work we're beginning in cancer, and then our pancreatic organoid, very similar to our artificial thymus, where we can create organoid for the pancreas that will excrete insulin based on glucose level of the blood. And I'll call up Robert Hoffman, our CFO, and he'll give you a quick overview of our financials, and then we'll turn it over to Hamid and get into the science.
So my one slide that I have for the presentation. So the company did a direct listing early last year, January 2024. We recently completed a deal with Yorkville, which $25 million in total committed. We did bring in $10 million gross before the end of 24 to help really buffer up, strengthen the balance sheet. So at the end of September, we had just under $8 million. Combine that with the $10 million gross, that with the planned activities of the clinical trial, which Hamid will tell you about, gets us into 2026. We did join the Russell Index, the Russell 2000, mid last year as well, but really pleased to see BlackRock owning 1.8 million shares. So that's above and beyond the kind of the index component. So they've really have increased their ownership, and we're seeing institutional ownership grow in that regard. Market cap roughly about $70 million. But happy to take any questions on the financial side after the presentation. I don't want to stand in front of Hamid, Dr. Koja, who's going to tell you all the exciting things that we're working on. So, Hamid?
Thank you very much, Robert. Thanks, everyone, for coming. We're going to go through our clinical product candidates, and it'll be a little bit science-heavy, so I apologize in advance for those who are not. So our first product pipeline in our pipeline is for diabetic foot ulcer. That is the CYC628. That is our product candidate for the treatment of diabetic foot ulcer. As Pete mentioned, that is due for a clinical trial this year, initiating at the end of Q2 of 2025. CYMS-101 is our product candidate for the treatment of multiple sclerosis. That has completed a phase 1 clinical trial, and I'll show you the data on that. It was a first-in-human, safety-only primary outcome. Cybersol is our product candidate for the treatment of degenerative disc disease. That has an FDA go-ahead for an IND. And then CYPS-317 is our product candidate for the treatment of multiple sclerosis. I will go in a little bit of detail in all of those in just a little bit. In early phase discovery projects, we have the CYTER915 for thymic convolution reversal, or an artificial organoid to recover the lost functionality of the thymus gland. TCB190 is our product for the treatment of cancer. We have several projects that are slated to begin this year. And then CYAP0913 is our product candidate for the treatment of diabetes using an artificial organoid. So let's go through wound healing, and this is basically almost all of our preclinical data and summarized for which are IND enabling. So diabetic foot ulcers, 6.3% of adults with diabetes, about 33 million worldwide, will develop diabetic foot ulcers. 20% will require a lower extremity amputation, of which 10% will die within the first year. 40% recurrence rate within the first year, and of course, another 70% recurrence rate in the same exact area within 3 years. So why fibroblasts for the treatment of diabetic foot ulcers? If you look at wound healing, any publications, there are a multitude of publications, you'll note that fibroblasts play a key role in every single stage of the wound healing, from the hemostasis to inflammation, proliferation, and remodeling. Just to note, fibroblasts are the key cells that secrete the extracellular matrix that maintain not only your tissues and your skin, but also in every organ in the human body. So what we utilize for our treatment for diabetic foot ulcers is not single cells. What we utilize is a spheroid of fibroblasts. These are basically about 3,000 cells. These are about 3,000 cells contained as a spheroid that are administered on the surface of the wound. They migrate out on the surface of the wound and release all the necessary factors such as cytokines and growth factors to initiate the wound healing process. Here you see topically administered on the surface of a wound, and here within 6 hours you see them migrating outwards. The reason why we use spheroids is, one, they're more viable than single cells. They don't require pre-culturing before administration. They can be frozen, thawed, and used. And second, they have a significantly higher potency and therapeutic efficacy than single cells. And we have quite a bit of data to show to prove that. Just showing a comparison between our product and product G, which is graphics from Smith and Nephew. And as you can see, within 20 days, about 83.8% of the wounds were healed completely with our product, as compared to about 66% with graphics. And these results are statistically significant. And it's not just being able to heal the wound quickly, which is of course important, but you have to be able to heal the wound with a high quality heal so that it doesn't recur. Because recurrence rates, as I said, 70% within 3 years, 40% within the first year, is significantly high for diabetic foot ulcers. So what we have done is we have monitored 7 different biomarkers which are key in determining a high quality wound heal. And I will go through those fairly quickly. We've been able to show that with our product, it promotes reepithelialization much better than graphics at day 19, which is at the remodeling phase of wound healing. Day 8 is at the proliferation phase of the wound healing. And as you can see, it's significantly better in terms of reepithelialization. Looking at granulation, which is often viewed by physicians as a sign of wound healing will occur, you can see significantly improved results as compared to graphics when it comes to granulation. We're looking at cell proliferation is important, right? Because what happens in chronic wounds is most of the cells, resident cells such as immune cells, such as fibroblasts and endothelial cells, they have become senescent, so they do not proliferate. So what we're doing is with our product, we are jump-starting and activating those cells again to proliferate. And here you can see both at the proliferation phase and remodeling phase, you're seeing a significantly improved cell proliferation as compared to graphics. Neovascularization. One of the major issues with diabetic foot ulcers is the lack of sufficient and efficient vascularization. if a vascularization doesn't occur, you're not going to heal the wound effectively, and it will not stay healed. So here we have been able to show that at the remodeling phase, we are impacting vascularization, and we're seeing significantly improved vascularization in the tissue that is being healed as compared to graphics. Fibroblast recruitment is an important aspect of wound healing process as well. And as you can see here, both at day 8, we're seeing a significant improvement of fibroblast recruitment, which initiates that wound healing process during the proliferation phase and then plateaus during those cells begin secreting extracellular matrix, etc., to continue the wound healing process. Keratinocyte migration, another important aspect of wound healing process. You want to be able to see keratinization occurring with keratinocytes proliferating on the surface of the healed wound. Here you can see significantly improved results as compared to graphics. Epithelial to mesenchymal transition, another important hallmark of a good quality healed wound. You can see that again, both at the proliferation phase and at the remodeling phase, we're seeing a significantly improved result as compared to graphics. Now, it's important to note that we are topically administering, but we seem to have a more, not only a local impact in terms of the cytokines and growth factors that are important in wound healing, but we seem to have even a systemic improvement in the immune modulation. And in here we're seeing, looking at serum level IL-6, which are important in the migration of leukocytes in the wounds, which is important at the initial step of the proliferation at about day 8, and then it plateaus. And you can see a significant improvement over graphics in terms of IL-6, both tissue and serum levels. We were not expecting to have a serum level improvement, but we're seeing a significantly improved IL-6. And we're looking at IL-1 alpha, which is an inflammatory cytokine, which is important at the proliferation phase, which leads to fibroblast recruitment. And you can see significant improvement at the proliferation phase, but then it goes away at the tissue level. It completely goes away during the remodeling step, but it's significantly high still at the proliferation phase. TNF-alpha, another inflammatory cytokine, and you can see that significantly high both at serum level and at the tissue level in these patients as compared to graphics. But then you can see by day 19 remodeling phase, the impact of this inflammatory cytokine has gone down, which is exactly what you want when you want to reach that state of homeostasis in the immune system, both systemically and locally, to initiate that tissue regeneration and wound healing process. IL-1 beta, again, another cytokine, very important in inducing fibroblasts and keratinocyte proliferation. You see those being significantly high at both at serum level and tissue level and at the proliferation phase, but then winding down at the remodeling phase, which is exactly what you expect when the tissue becomes goes through a homeostasis process in the immune system or infiltrating immune cells. IL-10, again, we see overexpression of IL-10 being very important in regenerating dermal wounds. There's lots of publications to indicate that, and you can see significantly high both at tissue level and at serum level with our product. So as a summary, you can see that we do have a platform technology. Our initial clinical indication is certainly diabetic foot ulcers, but this can be utilized for burn wounds. It can be utilized for surgical wounds, accelerating the wound healing process and surgical wounds, and it might have a retail component at some point as well. So it is a platform technology that we intend on moving forward. So in terms of toxicology, I will go through this fairly quickly. The cells do not graft. They go away within 4 days, as you can see with this data. We have some PCR results to illustrate that as well. They do not graft. They basically stay on the surface of the wound sufficient time, about 4 days, to initiate that wound healing process, and then they go away. And in terms of the impact on CBC, we see no impact on CBC, no impact on WBC, no impact in liver functions, and no impact in renal functions. So the safety profile is very good with this topical administration. So our next product candidates are both for autoimmune disorders, multiple sclerosis and psoriasis. The immune system is very intricate, right? So typically, it's all, for a healthy individual, it's at the homeostasis level. But then when you have an infection or you have an illness, it can go to the inflammation phase. When you're healthy, it always goes back to that homeostasis portion. But if you're not healthy, if you have an autoimmune disorder or you have chronic inflammation, it always stays at the inflammatory phase. And that causes significant damage to your organs and tissues. The problem is with patients with autoimmune disorder and patients that are suffering from chronic inflammation, they lack the mechanism to recover back to homeostasis in their immune system. And this leads to gradual cell, organ, and tissue damage over time. Now, with biologics that were introduced beginning about 11 years ago for the treatment of autoimmune disorders, which are also utilized for chronic inflammation, it introduced a completely another state for the immune system, and that's depletion. So what these biologics do is they either deplete an entire cell type or deplete the cytokines that are really important in modulating the immune system. So now you have a depletion state, and that's what but the problem is, when you deplete the immune system, you start initiating a whole set of adverse side effects. And these are, and you've seen these in commercials for these biologics that are on every night on TV. They always lead to compromised immune system. Viral, bacterial, fungal infections are wide. And some of them actually do have black box warnings in terms of complications with the cardiovascular and renal tissue. So what we're trying to develop is a potential for modulating the immune system back to homeostasis without depleting the necessary components of the immune system. And it's a unique cell-based, fibroblast cell-based therapeutic approach. It really does naturally guide the immune system back to homeostasis, and I will show you the data. It activates bodies, and which leads to the activation of your body's own natural healing or regeneration to heal whatever is wrong with your systems. And this would certainly lead to a potentially significantly reduced side effects that we see with some of these biologics. So we also certainly hope and imagine that this would be quite durable, as we've seen based on some of our animal model studies. So for multiple sclerosis, again, 2.8 million people worldwide, about 1 million in the United States, a very high prevalence in the United States, and the prevalence of multiple sclerosis worldwide has increased since 2013. And looking at our product, which is again a spheroid-based treatment injected into these animal models, and as you can see here in this animal model, we've been able to just with a single administration get rid of the symptoms of multiple sclerosis in these EAE rat models. And as you can see with stem cells, they go through relapse over time, but never recover fully. We were very shocked that we could have, with a single treatment, maintain and prevent this relapse. So when we look at the impact on the immune system, again, think about homeostasis and what's important. T regulatory cells are a significant key player in maintaining homeostasis in your immune system. And we look at, on the left panel, you can see in vitro data, on the right panel, in vivo data, we've been able to show significantly improved T regulatory cell expansion with our product in these animal models, as compared to both types of stem cells, both adipose and bone marrow-derived stem cells. And again, we've been also able to show that the reactive T cells in autoimmune disorders, which is often Th17, as you can see, both in vitro studies and in vivo studies, a significant reduction in Th17 and IL-17 in these animal models. Again, bringing those back to normal leads to homeostasis in your immune system. And again, we're looking at markers for anti-inflammatory cytokines and looking at IL-10, IL-1RA, and PD-L1, significantly upregulated anti-inflammatory response with our product for all these important markers of anti-inflammation. And looking at inflammatory markers such as CD40, CD80, and CD86, you're seeing a significant reduction in these inflammatory cytokines or inflammatory markers in these cells. We were also able to demonstrate remyelination. And we've seen that significantly improved as compared to adipose-derived stem cells. We've seen remyelination in these rats. And we were also able to see oligodendrocyte expansion. Oligodendrocytes are responsible for the formation of the myelin sheath. And as you can see, looking at two different independent methods of looking at myelination and oligodendrocyte expansion, we see those significantly improved with our product candidate. Yes. And looking at microglia. Microglia are the resident immune cells in the brain, and it is the microglia that their overactivity damages the myelin sheath. So when we look at two different markers specific for microglia, and you can see as compared to control here, you're seeing a significant activity of microglia here, and with adipose-derived stem cells, you still see that activity. With our product, you do not. And we've been able to show in other means as well, by other means, that microglia activity is specifically impacted and reduced. So as a summary, we've been able to show remyelination, which is something that others have not been able to show. We've been able to show an inhibition of dendritic cell maturation. Dendritic cells are the antigen-presenting cells, which are overactive in autoimmune disorders. And we've been able to show significant suppression of microglia activation. And our phase 1 clinical trial, first in human results, the safety profile was perfect. We had no adverse events noted during the injection of these fibroblasts. Again, first in human. No short-term or long-term impact noted in blood count during the 16-week monitoring period, and no short-term or long-term impact noted in electrocardiogram results. We included that because some of these therapeutics that are used for MS do have a adverse side effect in terms of cardiology. In terms of efficacy, two of the assays that we utilize that are utilized by neurologists to assess and gauge multiple sclerosis, we've been able to show significant improvement with all these improvement with these 5 patients. The other assays did not deteriorate, but they did not improve. Again, this was a first-in-human, safety-first clinical trial with 5 patients, so the results in terms of efficacy are not significant. We have to do a larger trial to find the statistical significance for the efficacy. Psoriasis, a wide range of impact worldwide. 123 million people suffering from worldwide. I'm sure there's a few of us in this room that probably have it. 7.5 million in the United States. It does have a significant lifestyle and psychological impact in patients. It is an autoimmune disorder that's mediated by both the innate and adaptive immune system. So we have looked at intravenous injection of our product. We saw very good results in multiple sclerosis, another autoimmune disorder. We wanted to see if it has an impact in psoriasis. We looked at 2 different variations of psoriasis, both moderate as well as severe. And here you can see, in looking at thickness, arrhythmia, and scaling, it's a PASI score. And looking at the average, we've been able to show with a single administration, in terms of moderate psoriasis, we were able to significantly improve the results of psoriasis. Looking at severe psoriasis, we've been able to show that with multiple administration, we can significantly improve the results as well of psoriasis. And looking at the data, we can see decreased thickening. Here's the control, healthy control. Here's the mocha mold, which is what causes psoriasis in these animal models. You can see the extreme thickening of the epidermal layer. Here you can see it much lower with a treatment of our product. And here you can actually see that it is that information is statistically significant. And you can see another assess here by keratinocytes. You can see in red, in thickening, you see an enlarged keratinocyte layer, and you can see it significantly lower with a single administration. And another problem with psoriasis is immune cell infiltration that leads to those raised plaques that are red and shedding. And you can see with our product, significantly improved results. So here's with a mocha mold, you can actually see quite a few immune cells. Here with our product, now statistically significantly better in terms of immune infiltration. And impact at a tissue level in terms of pro-inflammatory cytokines, you can see that being significantly improved. So again, at the tissue level, we're also seeing here significant improvement in bringing back that inflamed tissue back to homeostasis so that your own body could begin healing the plaques. We've also tested and compared against a monoclonal antibody for IL-23. That is the market leader for the biologics for the treatment of multiple sclerosis. As you can see, with a single administration, we've been able to show that it is significantly better than multiple administration of this monoclonal antibody against IL-23. So that's very important. A single administration, we've been able to show just as good of results as multiple administrations of IL-23. So we're currently testing multiple administrations to see how it would be better with our product. So as a summary, significantly reduced epidermal thickening, significantly reduced infiltration of the immune system, and so far, better results than a monoclonal antibody against IL-23. So just to go a little bit in our early research projects, that's for human longevity. That's developing an artificial thymic organoid for maintaining the immune system homeostasis. And I think Pete went in through this a little bit. The thymus gland is the teaching center for your immune system, and by the time it has about a 15.7-year half-life, so that means greater than 90% loss of functionality by the time people are about in their 60s and 70s. This leads to a reduced response to vaccination, increased risk of cancer, autoimmune disorders, and infections. So publications have indicated that 95% of the healthcare spending is for chronic diseases. And when your thymus is not functioning, your prevalence for chronic diseases increase. Okay? So what we've done is we've developed an artificial organoid that takes cells from the thymus gland and mixes them with our fibroblasts that are specific. And these are, as you can see, here's the spheroid. You can see the thymocytes in red and green and fibroblasts that maintains that spheroid functionality or that organoid functionality. It's very easy to scale up manufacturing. It is transplantation ready within 3 days. We've seen data that it fixates in the tissue for up to 60 days, and it vascularizes and continuously releases some of the important immune cells into the system. To just show you some data, it does give rise to T cells. We've seen that very clearly. Within 3 weeks of injection, we see the formation of immune cells. T regulatory cells, which are an important aspect of the immune system, which begins to falter as you age. That is significantly improved. And we also see gamma delta cells, which are very important in protection against pathogens and cancer. That is also increased. And in terms of the TCR beta chain, which is the recognition site, the variable recognition site that's important in being able to recognize multiple antigens, we see a very diverse chain as well. So that's very important. So we're able to recover the functionality in these animal models that don't have an immune system. We've been able to recover the functionality of the thymus gland. So again, transplantation ready, very easy to manufacture, very easy to scale up, and it does stay subcutaneously, it does stay and continue to pump out the necessary immune cells. We do have products that we're initiating some early phase projects for cancer. This is reprogramming the cancer-associated fibroblasts, which are an important aspect of maintaining the tumor and hiding the tumor from the immune system, and utilizing fibroblasts to deliver tumor inhibitory reagents, as well as inducing tumor vascular necrosis. And we're looking at making organoids that are based on CAR T and CAR natural killer cells as a potential vaccine and therapeutics. All right. So in summary, we have a clinical stage pipeline platform candidates ready to proceed to clinical trials. We have a very strong IP profile with 160 patents, 160 plus patents to date. We are issuing about a patent every 2 weeks. Experience management with over 60 years of experience, and we have completed the master cell bank, working cell bank, and the drug product should be ready by the end of Q2 of 2025 for our phase 1/2 clinical trial and diabetic foot ulcers. Thank you.